Sure. There's a couple of legs to my research. We specialize particularly in the microbiological classification and functional analysis and the human disease gene prioritization. So if I had to put a broad hat over the whole thing, I would say that function describes my research most precisely. Well, I guess my initial interest came when I didn't know what bioinformatics was. I went to school a while back and we didn't really have this flood of data that we do now. The first project that I worked on was building the components of the gene cards database. So this is a product of the Weizmann Institute, and I went over there on the recommendation from my advisor in State University of New York at Stony Brook, who actually hired me to do the analysis of just newly sequenced C. elegans worm genome. So I was coming out of that and going into this amazing place which was full of science, and I think it wasn't necessarily the project itself, which was fun to get to know all of this stuff, but how passionate everybody else was around this stuff. And from that point on, it just got better and better and better, and I can't believe how good it is right now. It's so interesting. The scientific take-home message is that we are not at the stage where we can make definitive predictions of complex disorders from genetic data. However, we are at the stage where we have enough data to make certain conclusions which could be followed up experimentally. So I had shown ways of finding genomic pathways which may be affected in individuals that have particular diseases. Specifically, I focused on Crohn's disease in this talk. And I think the other take-home message there is that what we believed 10 years ago to be the case, that the variants in healthy genomes are usually neutral functionally, is probably not true. So in fact, most of the diseases that we get are facilitated by having a particular non-neutral background in our genomes and then pushed over the edge by the environment. Oh wow, I don't think you can do much biology now without bioinformatics. There is very limited things you can do without analyzing your data statistically, computationally, looking at things, validating things. You mean the ones that I'm not addressing right now? There are many very, very, very good questions. So one is, what is this hidden genome regulatory region story, right? So Yanas talked this morning on about these transcripts, which are not canonical transcripts. And actually the field is very much divided about how many of those there are and how significant there are. If you listen to just his talk, then you would come out thinking, okay, the normal standard canonical genes don't really matter. They make up less than 10% of the population or something like this. And if you listen to other talks, right, that completely ignore, including my own, that completely ignore the non-canonical splice regions, splice genes. So it's not that important. So I don't know what the answer is, and when you don't know what the answer is, that's a really great question, and I think bioinformatics is prime. Well, I'm a bioinformatician. So rapid change in bioinformatics. And we are a new field, we're growing rapidly, you're right, but I think we are acquiring the characteristics of a stable field where, and this is a good thing, where reproducibility actually matters. And I think that in the future, it's actually going to get to a point where we have a standardized set of methods that we use, where we're developing tools that biologists are comfortable using, know how to use, understand how to use. And hopefully the new generation of scientists doesn't need this prodding that you should be using bioinformatics, and they understand how to do it themselves. Do things that don't sound like you can do them. The more you try, the more you learn, and no knowledge is ever wasted. So that, I think this was the advice of my grandfather. No learning is ever wasted, and I go by that every day. Do the things that you do not know how to do, you think you do not know how to do, and you'll be surprised how much you can get done.